Human immunodeficiency virus has infected around 37 million people around the world, with sub-Saharan Africa most affected. Populations at risk include men who have sex with men, injection drug users, and sex workers. HIV is transmitted via bodily fluids, where the virus must enter the bloodstream for infection to occur. The envelope surrounding the HIV contains a membrane protein complex consisting of extracellular GP120 and transmembrane GP41. The ENV gene is responsible for encoding these proteins. On contact with the host's CD4 helper T cells, GP120 binds to CD4 and co-receptor CCR5. GP41 then initiates fusion of the virus into the cell via endocytosis. Once inside the host cell, the viral RNA is then able to replicate and produce more viral particles. These particles bud off of the host cell, lysing it, causing a decrease in CD4 effector helper T cells that orchestrate the immune response. To combat against invasion, the immune system initiates an adaptive immune response. Antigen arrives through the afferent lymphatic into the subcapsular sinus, where they are broken up by macrophages, which are then processed and presented to naive B cells within the follicle. The naive B cell binds to an antigen and receives a secondary signal through T cell mediated release of cytokines, allowing it to proliferate, switch isotype, and affinity mature. This process creates effective plasma cells that secrete antigen-specific antibodies. HIV has an extremely rapid mutation rate, and as a consequence, efforts to produce a vaccine have been inconclusive. Vaccination is the deliberate exposure of the body to a particular pathogen using either an inactivated, live attenuated, or non-pathogenic form of the pathogen to induce immunological memory. We are proposing a subunit vaccine where the ENV gene is inserted into a modified yeast organism where the GP120 and GP41 proteins will be harvested. The harvested proteins will then be bound to a liposome in order to retain the conformationally stable state and function of the membrane protein. These liposomes are then injected intravenously into the patient to mimic an HIV infection as it spreads throughout the body from the blood. Two adjuvants, ASO1B and ALFQ, will also be injected with the vaccine to enhance immune responses. They provide long-lasting immune responses for up to 18 months and enhance helper T cell responses, respectively. In order to induce an immune response, inflammation will also be induced by the addition of cytokines. Our vaccine will induce a primary response by exposing the body to the conserved proteins GP120 and GP41 for the first time. An individual is able to make antibodies against these proteins without exposure to the HIV virus. Over time, individuals will begin to build resistance to these proteins, strengthening immunological memory. The goal of this vaccination is to allow the patient's body to produce broadly neutralizing antibodies. This can be done by exposing the antigen to the body for prolonged periods of time. To achieve this result, we intend to administer the vaccine every 3 months for 2 to 5 years. Every 3 months, we introduce a new variation of epitopes in an effort to help produce those broadly neutralizing antibodies. This also ensures that the liposome remains intact at all times. The dosage of the vaccine should be enough to overwhelm the innate immune response and allow the onset of adaptive responses. Over time, the number of virus-specific antibodies present in the blood eventually plateau. Multiple exposures will increase the strength and speed of the immune response to future exposures. Various approaches have been tested for developing an HIV vaccine, but many studies have had an issue with the lack of immunogenicity due to the virus having a rapid mutation rate. Because of the inability to generate strain-specific neutralization, and only a limited number of approaches have emerged, the body is exposed to GP120 and GP41 for a longer period of time and more frequently, allowing development of broadly neutralizing antibodies. Other vaccines were administered every 6 months for 3 years, allowing the virus more time to develop new strains not recognized by the immune system. Our vaccine is administered every 3 months for 2 to 5 years, to ensure new variation of epitopes are being introduced more frequently. Continuous exposure of HIV proteins delivered through the vaccine will provide the body with a growing repertoire of HIV-specific antibodies.